Welcome back to Dr. Vipin's Biotech and Bioinformatics classroom. And over the last few lectures, I have been emphasizing the importance of programming in biotechnology. Like all other fields, biotechnology too is under transition, and it is fast becoming a data-driven science. And this is largely because of the omics revolution. So we are now mostly dealing with high throughput techniques, and therefore the data that comes out is large in volume and large also in variety, and therefore it is not manually possible to analyze such data, and that necessitates that a certain level of programming expertise is there in the candidate to analyze the data. However, one important thing is that programming is not everybody's cup of tea, and also it takes some time to acquire the skills of programming. Not every biologist is trained to write a code, and therefore you may not have enough time. And some of you may be into your PhD final year or third year or beyond the second year, and you may not have enough time to actually start learning coding and then implement it for data analysis. So for such users, there is still hope, and that hope is in the form of the workflows that are available to you. One such common workflow is Galaxy. So what is Galaxy? Galaxy is an open source web-based platform for accessible, reproducible, and transparent computational biomedical research, right? Very importantly, it allows users without programming experience to easily specify parameters and run individual tools as well as large workflows. So two important things. One, Galaxy is open source, which means it is available to everyone for use. It is free of charge for academicians. And second important point is that it is based on FAIR principles. FAIR stands for findable, accessible, interoperable, and reusable data, right? So there is a certain quality of data that has to be there when you do your research, and all the scientific research is now based on FAIR principles. And you can have a detailed look at FAIR principles from this website here, right? So coming back to Galaxy, Galaxy was initially launched in 2005. There is a genome research paper for the Galaxy. And based on the feedback and other requirements and also the changing technology, there are regular updates on Galaxy. And this is the 2020 update report from Nucleic Acid Research. So one more time, what is Galaxy? Galaxy is an open source platform for fair data analysis that enables users to use tools from various domains that can be plugged into workflow through its graphical web interface. And we'll look at the web interface very shortly, and you'll find that this is really comfortable and very easy to use. It is more like a graphic user interface to all the tools that you use in NGS data analysis or protein structure function analysis, right? Then, of course, you can run code in interactive environments using RStudio, Jupyter, along with other tools and workflows. You can manage data by sharing and publishing results, workflows, and visualizations. So what is also important is you could create a workflow once, and then, of course, you just need to feed in your new data, and exactly the same steps will be followed, and the final results will be compiled. And this workflow is shareable between individuals, so that makes it even more handy, right? And then, of course, Galaxy ensures reproducibility by capturing the necessary information to repeat and understand data analysis. And there is a very active Galaxy community. You can write to them in case you get stuck anywhere. They also develop protocols and prototypes of how to solve a particular type of problem using Galaxy interface, some of which we'll also do in this track. The important point here is that to use Galaxy, you do not require any prior programming knowledge. And that makes Galaxy a very important platform for people who are struggling to code or who would want to learn code over time, but may not have time enough for now to experiment with learning the code. So what we're going to do in this lecture is to have a look at the interface of Galaxy, and parallelly we'll do the same exercises in command line in the Linux environment. So let's get back to Galaxy, and here is the Galaxy main server. So this is called usegalaxy.org, and in case you're logging in for the first time, you may have to register first, and then you can start using the Galaxy server, right? So once you log in, this is the interface that is going to open up. There are three compartments in this interface. On the left-hand side, you have the tool shed and also the option to upload the data. In the central panel, you'll be able to see the data analytics and the results. And then in the right-hand panel, you have the history. So basically, it shows you what are the previous projects that you have run here, and the latest one would be shown up. For example, here in this case, you can see this is MRSA Nanopore. So this is an exercise that I've done the last time. And then, of course, it is showing the steps of those exercises here. And the ones in green basically mean that these steps were successful, right? So all steps that you do here will be lined up here. So there are some 22 steps in this exercise that we have done, which are being shown up here. So let's start with the fresh exercise. So here we are, we can go to, create a new history. So you say plus, and you create a new history. So this time you can name this history. So let's say we name it as galaxy underscore introduction. Introduction. Right? And you press enter. Right. So this is now my fresh project, and this is called Galaxy underscore introduction, where I'm trying to introduce you to Galaxy. So as I said, there are three main compartments. So on the left-hand side, there is the tool shed from where you can select the tool that you want to use. Also, you can upload your data through the upload data option here. Likewise, you could also download your data from here. So if you click on get data, it will ask you to download options will be available to you, right? Then, of course, on the right-hand side, you have the history panel, which shows you the exercises that you've done earlier. Also, it allows you to create a new history, which is your current program. And then, of course, the processes that you do in this particular history will line up here as we move forward. So let's first upload some data. And we're not practically going to do the exercise, just to give you an idea of how to upload the data. So here you are, you can go to upload data here. And there you see, you have the options of choosing the local files. You also have the option of paste or fetch data. That is to basically use the URLs to download the data. So we would use the choose local files option here. So we go back here and choose the local files. So here I'm choosing two FastQ files from the files that I already have. So if you see here, there are two files with the same name, SRR 2589044 underscore 1. And then, of course, you have the second file that is the same name, except that it has an underscore 2 indicating that this is basically a paired and read data that we are uploading here, right? So you say open and you click on this, and that would then allow the files to be loaded here. So if you see, it says you added two files to the queue, add more files, or click start to proceed. So now we click start and the files get uploaded here, right? So here you are, the files are getting uploaded, and you can see the bar that indicates the progress. All right, so here we are, and our the paired and read for the other end is also uploaded now, and it is currently orange, and it will turn into green shortly, right? So here we are, we can close this now. We have had complete uploading, so we could close this. And here we are, this is still getting processed, and now it is turned green, which means that the files are uploaded. Now you could have a look at your files, a sneak peek preview from here itself. So you double click, and there you are. So if you see here, this is format is FastQSanger, database is question mark, uploaded FastQSanger file, and then of course you can have a sneak peek of your files here as well. Likewise, you could click on this one here. That will also give you the idea of format FastQSanger database. And then, of course, it gives you some idea of this. So if you want to have more overview of your file, you could click on the I button here. This will open your file in the central panel here and give you an idea of what has got uploaded here. So here you are, the data set is large, and only the first megabyte is shown here. So if you see here, you can click on show all to see everything. So this is typically your FASTQ format file. You can see a typical 4 4-line format. The first line begins with the @ sign, and then it has the platform information, also the flow cell information. This is the unique identifier. The second line is the actual sequence. The third line begins with the plus sign. In most cases, it will also have the identifier repeated. But here in this case, you only have the plus sign and no further information on this third line. The fourth line is the FASTQ quality line. That is basically to indicate the quality of the base call at that particular position. And this is ASCII encoded. It is not in terms of absolute numbers. Then if you move on, you would, if you want to edit something, you could click on edit attributes. So here you are. If you click edit attributes, you will come up here where you can have the attributes of the file. You could convert the file into a different format if you want, depending on what is the requirement for the tool that you want to use downstream. And then, of course, you could also toggle with the permissions of the file, right? Now we come to the process. So let's say you want to put up the files to FastQC or quality control. So FastQC is basically a program that comes from Barbraham Lab and basically allows you to have the first view of the quality of your data. In terms of the Phred score at each position, also it allows you to look at the several other parameters, for example, the adapter contamination, GC content, and so on and so forth. We will do a more detailed video on FastQC later. This one is more to show you how you can pull out the tools in Galaxy. So here you are. This is your tool shed. Now that you've uploaded the data, you could search for the tool that you want to use. Let's say we want to use Fly, FLYE, right? So FLYE. You press enter and it will basically give you the Fly tool here. If you double click, now the interface for Fly input will come up. We are not going to use Fly, of course. I'm just giving you as an example. So Fly is a de novo assembler for single molecule sequencing reads, right? Likewise, you could search for, let's say, star. So you could give the exact term, or you could give the, the term that is part of your tool that you want to use. So when you put up star, there are several tools that have star come up. For example, RNA star solo. This is for mapping, demultiplexing, and gene quantification for single cell RNA-seq. Then you have star fusion, detect fusion genes in RNA-seq data. RNA star, this is a gapped read mapper for RNA sequence data. Then you have star AMR. This is basically a program that scans genome assemblies against the res finder, plasmid finder, and point finder databases to search for antimicrobial resistance genes, right? You could also type the process that you want to do, and that will again fit you the tools that are related to that process. For example, I could type ASSEMBLY, and this will give us a lot of assemblers that can be used for my purpose. For example, here you have the medical consensus pipeline, assembly polishing via neural networks. Then you have GFA to FASTA, converts graphical fragment assembly files to FASTA format. Mini ASM, ultra fast assembly for long noisy reads. Then you have QAST, this is genome quality assembly, genome assembly quality. Then you have bandage for determining the statistics of de novo assembly graphs. Likewise, you have Mercury, evaluate the assembly quality. Racon, consensus module for raw de novo DNA assembly of long uncorrected reads, and so on and so forth. You have a whole list. So basically, either you know the name of the tool that you want to use, or if you want to scan what are the tools available, you could also give up the actual process that you want to do, and it will suggest tools accordingly. So let's now move forward and do a fast QC and see what do we have here. So let's say you want to do a fast QC. So we could search for fast QC now. So here you are, you say FAST, fast QC, right? And here you are. So this was your read quality reports, fast QC read quality reports. So you could click here, and now this is the page where you need to upload your data for fast QC. So whatever tool you select for, its graphic user display for uploading of the data would come up here, right? And so what we are going to upload is raw read data from the current history. So we click on, and since we have two files, we click on multiple data sets. And we select these two files, underscore two and underscore one. And then of course we leave everything else as default. And we say run tool. So you could come up here and you could click on run tool to run your tool. This will then take this two files as input to FastQC and basically do the quality control and give you the quality control report. So here you are. Let me just run the tool here and see what comes up. So if you see now, you are showing the processes. So each file will generate two reports. One would be raw data, the other will be the web page. So finally, if you see all of these have turned green, which means that the process is complete, and you can have a look at the results. You have two types of results here. One is the raw data, the other is the web page. So let's have a look at what you get in the raw data. So we can look at the underscore one file, that is your forward primer sequencing reads. So we look at the raw data first. Here we are. So this is your raw data for file one or the forward read. This is SRR 589044 underscore one dot fastq. And then of course it says that there are total number of sequences 1107090. Sequence quality flagged as poor is 0. Sequence length is 150 bases. Percentage GC is 50. And then of course you have the statistics. So you have the upper quartile, the 10th percentile, the 90th percentile, and so on and so forth, right? So there's basically the details of the box plot and the other details which we'll discuss when we discuss FastQC in details the next time, right? Let's also have a look at the web page. So for web page, we can look at the second or the reverse primer files. So here you are, you can click here. So this is basically your report that is majorly obtained from FastQC file. The most important graph is the per base sequence quality graph. And here, as you, if you see towards the end of the read, you have very poor quality data because the distribution of the scores ranges from 2 to as much as 34. So higher the score, the better it is, but the lower ranges are also extreme here. So you have some of the reads at this position of 135 to 139 are in the range of 2, 3, or 4 scores, which are very poor, right? So this is typically, it can suggest that your quality of read depreciates as we move towards the end of the read, right? So this is basically something that we need to keep in mind. So this is typically a poor quality read sequence data. So this is typically a poor quality sequence data, and as you can see, it is also indicated by the cross mark in the report, right? So this is something that needs to be corrected, and therefore downstream when you move on for the quality correction, you could use Trimomatic to trim parts of your sequence, right? So very clearly, you could easily trim the from at least base 120 to 150 can be uh, straight away cut off from here, right? So anything above 28 is a good score. Anything below 20 is an absolute no-no, and you can see there are whiskers that come below the 20 mark here and from position 75 onwards. So which means that you need to do a significant cropping of these reads before you can take it forward for the next analysis. Then there is a whole lot of graphs that you get here, and you can see very clearly 1, 2, 3, 4. So at least on 4 parameters, the quality control fails, and therefore you need to take measures to control these failures, right? So what you can very clearly see is that this is poor quality data, right? Let's also have a look at the score 1 to see if it is as bad a quality or better. So we could look at the underscore 1 web page. Here you are. Unfortunately, this one is quite good. If you see here, there is whiskers come until the end, but then mostly the data is concentrated in the range of 38 to 28, right? Which is very good quality. Anything above 30 is extremely good. Anything below 20 is not to be considered, right? And then, of course, other general parameters are also good here. So we'll discuss what each of these graphs mean in the next lecture. So I hope I have given you a fair idea of Galaxy, and you can make a start now. And then, of course, as we go through the track, we'll load more lectures so that you get more convenient, more comfortable with Galaxy. So this is one way where you can side by side keep learning your programming the hardcore way, Python or R. And alongside, you can also keep progressing with your work, right? Of course, knowing the programming is going to be of much help. But in case you don't have a knack for programming or you're not making enough progress, Galaxy and other similar workflows are a big help. So on that note, we close here, and I see you again with the next lecture, where we'll do a more formal introduction to fast QC and NGS data analysis. Take care, and please do share. Thank you very much, and I see you again for the next lecture very soon.